Hello everyone, welcome to another episode from Chedexcel. Today we are going to talk here about the different methods we can use to measure the rate of a chemical reaction. Now the last video we discussed about the catalyst, and I told you there are two types of catalysts called homogeneous and heterogeneous, and we explained and I explained there how each catalyst behave to speed the rate of the reaction. So you can use the link or you can use this pop-up banner to go to that video and get the idea about a catalyst. Now we are continuing off the unit 4 in Chedexcel syllabus. So rate of a reaction is simply defined as the change in concentration of time. So rate is defined as like this: change in concentration time taken. So that is so that's why you have the units of mole per decimeter cube divided by time seconds. So mole per decimeter cube, you get mole per decimeter cube per second. So that is the unit for the rate of a chemical reaction. Now even though we say the rate is a change in concentration for a given time period, it is practically impossible to measure the concentration change all the time, because not all the chemical reactions happens in the same way. Sometimes they have a color, sometimes they make a precipitate, sometimes they make a gas. So measuring the concentration change for a given time period is not practically possible all the time. So what we are going to do when measure the rate of a reaction is, depending on the chemical reaction, we use a specific method for that particular reaction, and later we convert the whatever the thing we find for a given time period into the concentration change. For example, there are chemical reaction which produces a color. So we can measure the color intensity and later we convert that color intensity to the concentration change. How do we convert the color intensity to concentration change is not in our syllabus. So you do not have to worry how we can do that one. So the idea is, we measure a different factor. It might be the temperature, it might be the concentration, it might be the gas volume, it might be the mass of a precipitate for a given time period, and later then we use that particular substance or that particular quantity to measure the rate of the reaction. Now as it is change in concentration or time taken, whatever the factor we can measure with respect to the time as well, which means let us say we are measuring the gas volume rather than the concentration change. So we can measure the gas volume for a given period of time, or we can measure the time taken to give the certain volume of the gas. So both ways are fine when we are measuring the rate of a reaction. So let us see what are the different types of reactions and one by one how we can use them to measure the rate of a reaction. Right. Let us say if the reaction produces a gas, these are different methods. We can measure the gas volume, right. Commonly they have asked as well, we can use something like this kind of apparatus setup to measure the gas, right. Normally we keep it in a water bath to keep the same temperature. This is the thermometer. This one is the gas syringe. So this is the gas collected. So we can measure the time taken to produce a certain volume of a gas, or we can measure the volume produced for a certain time period. For example, we can use this kind of setup to measure the rate with this reaction. Calcium carbonate reacts with HCl to produce calcium chloride plus carbon dioxide and H2O. Carbon dioxide is a gas. So in order to measure the rate of this particular reaction, rather than measuring the concentration change, we can measure the time taken to produce a certain volume of carbon dioxide, or we can measure the volume of carbon dioxide produced for a certain time period, right. So if the reaction produces a gas, we can use this kind of reaction and this kind of apparatus to do so. So one thing is measuring the gas volume. The second one is number 2, we can measure the color intensity, or we call it as a colorimetry. right. We can measure the number 2 as colorimetry. Let us say we have a simple reaction, let us say 2 Ki reacts with Cl2 to produce 2 KCl plus I2. Iodine has a brownish color in water. So, with the time being we can say the color intensity would increase. So, rather than measuring the concentration change of the reaction, we can measure the color intensity of iodine using apparatus or the instrument called colorimeter, not calorimeter, colorimeter. There are small quotes like stuff you put, I think you get those stuff in biology. So, colorimeter or the colorimetry we can use to measure the rate of reaction if the reaction produces a color, that is number 2. Third one, if the reaction produces a precipitate, we can measure the mass of the precipitate, right. So, we can measure the third method, we can say measuring the mass of a precipitate. We can measure the mass of a precipitate, or we can check the time taken to produce a certain mass of the precipitate. Now, when we measure in the time taken to produce certain mass of precipitate, what we do is, the common reaction is the reaction with sodium thiosulfate with HCl. Sodium thiosulfate reacts with HCl to produce sodium chloride plus sulfur dioxide plus sulfur and H2O. Due to the production of this sulfur precipitate, right, what we do is we allow the reaction to happen in a conical flask, and we keep that conical reaction mixture or the conical flask on the black colored or X. We make a mark across using a pen, and we put it our beaker on the top of that mark, and we do the reaction and we measure the time taken to disappear this cross, because formation of sulfur like a cloudy precipitate, the cross that we have placed get disappear. So, we can measure the time taken to disappear the cross with the different concentration of sodium thiosulfate or HCl. So, rather than measuring the concentration change, we can measure that one as a method to measure the rate of a reaction. So, that is how we do when we measure the mass of a precipitate, or when we measure the time taken to disappear or time taken to produce a certain mass of the precipitate, right. So, we can assume if the same person is doing this one, his eye sensitivity depends, so the same person should do this reaction. So, the time taken to disappear the cross will normally give the same mass of the precipitate. So, we can get the idea how fast the reaction or how slow the reaction is, right. That is the third one. The fourth one, we can measure the pH of the solution, right. You can say fourth one as measuring pH values. We can measure the pH values. If the reaction has an acid or a base, we can measure the pH value during the reaction. So, the pH value will give the idea how much acid or base is formed or how much it is being used up during the process. So, we can measure the pH value for a certain time period, or we can measure the time taken to give a certain pH change. So, that thing we can use to measure the rate of the reaction at the end, right. So, let us say acid is involved in the sodium hydroxide with CH3CH2Cl, CH3CH2OH and NaCl. We can measure the pH because sodium hydroxide is a base. So, with the time being sodium hydroxide being used up, so we won't be able to see, we will be able to use the pH measurement to measure the rate of the reaction. So, that is the fourth one. Other than that, fifth one we can say like this, we can measure the conductimetry, electrical conductions, right. We can measure the electrical conduction. So, that is also another method we can use. If the reaction produces ions, we can use electrical conductance to measure the rate of the reaction. right. So, those are 5 common methods that we can use. Other than that, the usual thing we can do, which is the titrations, right. Titration is commonly used to measure the concentration change itself. So, we can use a titration to measure the concentration of that particular instant. So, that is also possible, but compared to the other, that thing have more work to do, because we have to get the samples out, stop the reaction and measure the rate by measuring the concentration. So, time to time we have to do a titration, but still it is possible, right. So, those are 6 different methods that has commonly asked. Now, there are some other additional details you should remember in this part. One thing is, there are some methods when we measure the color intensity or the color change of the chemical reaction. There are sometimes we measure, okay, we can write it like the 7th point, measuring color change, measuring the color change. Actually, what we measure is the time taken to give a certain color change. Now, when we measure this color change, commonly this comes when there is a bromine and when there is an iodine, we have to use an additional substance in order to delay the color change. It is a very common question they always ask. For example, when a bromine is produced, we use methyl red indicator, because bromine reacts with methyl red indicator to give the color change from red to colorless or brown to colorless. But depending on the due to the reactivity, even one single drop of bromine is present, it gives the color change. So, we will not be able to measure the rate of the reaction, because no matter whether the bromine amount is low or high, the reaction happens very quickly. So, we would see the color change. So, let us say we are getting bromine from bromide. So, no matter what the bromide ion concentration, we get the color change very quickly. So, in order to delay that thing, what we do is we add a known volume or known amount of phenol. So, phenol would react with bromine first, and only after all the phenol is being reduced or removed, the bromine give the color change. So, no matter whether bromine concentration is low or high, we can't measure the rate. So, what we do is we add a known volume of phenol. So, phenol reacts with bromine. So, when the added bromine, when the added phenol is completely removed from the bromine, then only bromine gives a color change with methyl red indicator we have added. So, that is a very common thing. So, phenol is used just to delay the color change, because phenol effectively react with phenol, bromine effectively react with phenol first, and only after all the phenol is removed, it gives a color change. The other thing is, sometimes the similar concept is used with the iodine as well. The iodine normally reacts with starch to give the blue-black color. So, in order to delay that color change, what we do is we add non-volume or non-amount of sodium thiosulfate. So, only after all the sodium thiosulfate is removed, then only iodine gives a color change with starch. So, we use non-volume of thiosulfate just to delay the color change. Those two have commonly asked. When we discuss papers, we can come back to that again. Now, to measure this rate using those above methods, either we have to take samples out or we can use the whole solution at once. If you take the samples out, we say it is a sampling technique. For example, if you want to do a titration, if you are going to measure the color intensity, we have to take the samples out, because we can't use the whole solution at once to do a titration. If you take the samples out, then we have to stop the reaction at that particular instant, and we have to measure whatever the quantity we are going to measure. To stop the reaction, we say to quench the reaction, we use ice water bath. So, using ice water bath, we can quench it, and then we can measure the whatever the quantity we are going to measure. Especially that method is used to measure the to do the titration or measure color intensity. If we don't have to take the samples out, like if you are going to measure the pH value, if you are going to measure the volume of the gas, we don't have to stop the reaction. We can use the whole solution at once and we can measure it. So, that thing we called as continuous method. Actually, continuous method is much easier than the sampling method. So, continuous method, we don't have to take the samples out, we can measure the quantity for a given time. Now, both methods, normally what we have to do is, when you are measuring the rate with respect to one reactant, we have to keep the concentration of the other reactant to very high value or to make it a constant. Rather than making a constant, we can keep it very high value. So, the change or the using of that particular reactant is negligible. So, either we can make the both reactant to the constant, either we can make the reactant to a constant value while changing the concentration of the other reactant, or we can change the concentration of one reactant and keep the other reactant relatively very high value, like 1000 times higher than the reactant we keeping the concentration 1000 times higher than the other, than that one, we can make the reaction to go a normal way. We do not have to keep it constant, because if the concentration of the second reactant is 1000 times higher, the usage is negligible compared to the other reactant, right. So, those are the methods we can use. So, the next video we will be talking about how we can actually use those methods to measure the rate of the reaction and to measure something called the rate expression of a chemical reaction, right. So, if this is helpful, give us a thumbs up and consider subscribing Chedexcel to learn how more and more about chemistry and to pass your Chedexcel exams really well. Thanks for watching.